There. Okay. Hello and welcome to today's Journal Club featuring Zhen Yan. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research, news, and analysis. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation, but you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this journal club if that material has already been published elsewhere, or if you have first obtained express written permission from the presenter. Professor Zhen Yan joins us today from Buffalo, New York, where she is SUNY Distinguished Professor of Physiology and Biophysics at the State University of New York at Buffalo. Before joining the faculty at Buffalo, Professor Yan completed her PhD in neurobiology at the University of Tennessee under Dr. James Surmeyer. She then went on to a postdoc position at Rockefeller University before coming to SUNY Buffalo in 2000. Professor Yan has garnered numerous awards in recognition of her work throughout her career. Her lab's research goal is to understand the synaptic mechanisms commonly involved in the pathophysiology of a variety of brain conditions, including autism. The Journal Club today will focus on her recent paper published in Nature Neuroscience. The paper details a novel treatment for social deficits in an autism mouse model. Welcome, Professor Yan.
Thank you. Thank you for attending the Journal Club. And today I will talk about an epigenetic mechanism-based treatment strategy for the social deficits in Shank3 deficient mouse models of autism. So first of all, what is epigenetics? Epigenetics most often denotes changes in the chromosome that affect gene activity or expression without the alteration of genetic code itself. For many brain disorders, the pathogenic mechanism that is common usually involves the genetic risk factors and environmental factors. The combination of these two factors will lead to the disease-specific epigenetic changes. And then that will lead to the pathological genetic alteration, and then finally affect the synaptic function and the behavior outcome. So this is not only happening in autism, but also potentially in other developmental disorders, psychiatric disorders, or neurodegenerative disorders. So you can see that epigenetics is mediating the gene and environment interaction as a kind of a bottleneck. So first we would like to talk about autism. So as you all know, it's a very prevalent neurodevelopmental disorder characterized by the impaired social interaction, repetitive behavior, deficits in language, and decreased cognitive activity. So far, there's actually no treatment for the core symptoms of autism, such as social deficits and repetitive behaviors. Many autism risk factors have been identified from genetic screening. And among them, one of the causal factors is the Shank3 gene. The heterozygous deletion or loss of function mutation of Shank3 has been found in about 1% of all the autism spectrum disorders, or 2% of those with autism spectrum disorder and intellectual disability. So Shank3 gene is located on chromosome 22q13.3. The Phelan-McDermid syndrome, which is also called 22q13 microdeletion syndrome, has a high chance to have the autism. In fact, actually 75% of those patients have autism and 85% have autism spectrum disorder. So it's a highly penetrant gene for autism. Because of this reason, many mouse models expressing different mutant forms of Shank3 have been generated, and these mouse models exhibit different autistic-like symptoms and synaptic dysfunction to a certain extent. Shank3 belongs to a family of Shank proteins, and they are scaffold protein in the synapses. The The deletion or mutation of Shank3 has been linked most closely to these diseases. At a young age, they tend to develop autism spectrum disorder, intellectual disability, ADHD, etc. At young adolescent or at young adulthood, they tend to have a higher chance for schizophrenia and bipolar disorder. At the elderly stage, they may have Alzheimer's disease. So Shank3 has been implicated in a variety of disorders. To study why Shank3 can cause autism and how to treat core symptoms in these Shank3 deficient mouse model autism, we used a mouse model that has this heterozygous deletion of C-terminal, which is also exon 21 of Shank3. So Shank3 has many domains. The exon 21 contains important binding region for Homer, which is an anchor protein for metabotropic hormone receptors, and cortectin, which is an actin binding protein. We already demonstrated that in these heterozygous Shank3 C-term deletion animals, they have the loss of the full-length Shank3 protein, and the truncated protein, the C-terminal truncated protein, can be found in the total lysis, but not in the postsynaptic site, the PSD fraction, suggesting that when the C-terminal Shank3 is lost, Shank3 is no longer located at the synapses. This has been proven by multiple antibodies against different regions. This is against the Shank3 SH3 region, this is the PDZ domain, and this is the C-terminal. So they all demonstrate the strong loss of the full-length Shank3 and the loss of the synaptic localization of Shank3. To test whether these Shank3 deficient mouse model have the autistic-like social deficits, we use the 3-chamber social preference test. First of all, the animal is habituated in an apparatus that contains 2 empty capsules, and the animal is allowed to explore each of them. And then, during the test phase, the first phase, we put 2 identical non-social objects in each of the capsule, and the animal is free to explore each of them. And then in the second phase, we replace one with a social object, which is the same strain, same sex, same age animal. Usually it's a Y-type animal. And then the other one, we replace it with that novel non-social object. And then the animal is allowed to explore or investigate each of them. And the third phase, we introduce a new social stimuli. So the animal can either interact with the novel social or the familiar social. The phase 2 is the most important test because it will tell you whether the animal is preferring the social stimulus versus the non-social object. So from this study, we found that in the Y-type animal, they spend dramatically more time interacting with the social stimulus compared to the non-social stimulus. However, in the Shank3 C-terminal deleted heterozygous young male mice, the interaction time on the social object is significantly reduced, and the preference, the social preference, is also strongly decreased. So when you calculate the preference index, the second phase, the social versus non-social, you can see the strong decrease of the social preference in this Shank3 mouse model. The preference index basically just use the time in investigating the social minus the time investigating the non-social and divide by the total time investigating both. So this is a very robust social preference deficit demonstrated in this kind of Shank3 mouse model. Another sociability test is called social engagement or social approach test. Basically, you put a social stimulus under a capsule and then put the test animal in this apparatus and see how long this animal will interact with the social stimulus and how many times they approach that. And when we calculate the total sniffing time, we also found that these Shank3 deficient mice have strongly decreased investigating time. So these animals definitely have social deficits. Then we first want to know what the potential molecular target that is mediating these behavior deficits. So we turn to the Shank3 interacting proteins. And as I mentioned before, Shank3 has several important regions. The protein-rich, the Exon 21 region, has the homobinding site, which is anchoring protein for metabotropic receptors, but it also contains the cotectin binding sites, that is interacting protein with actin cytoskeleton. And through the PDZ domain, Shank3 can also interact with NMDA receptors. And then another thing is that we need to know which brain region to look at. We focus on prefrontal cortex because we think that this is a region highly involved in autism. There are several reasons for that. First of all, prefrontal cortex controls the high-level executive function, such as decision-making, emotional control, personality. All these things are impaired in autism. Secondly, the genetics analysis of autistic brain have found many genes that are differentially expressed or altered in this area, like 444 genes, but only 2 of them were found in cerebellum. So obviously, this region has a very important involvement. And then in the anatomic studies of autistic brain, people found focal patches of abnormal organization in prefrontal cortex, and the clearest deficits actually exist on those layer 4 or 5 excitatory cortical pyramidal neurons. So those are the pyramidal neurons we studied in electrophysiological recordings. And what we found is that the NMDA receptor-mediated excitatory synaptic responses are significantly reduced in Shank3 deficient animals. In contrast, the AMPA receptor-mediated excitatory synaptic current was unchanged. When you look at the NMDA to AMPA receptor EPSC ratio, it's also strongly reduced because of the loss of NMDA receptor function. And we further did biochemical studies to try to figure out how come the NMDA hypofunction occurred in these animals. And we found that in the total lysis, these receptor subunits have normal expression, but in the synaptic membrane, the NMDA receptor subunits NR1 and NR2A had a strong reduction. So it suggests that NMDA receptor trafficking to the synaptic membrane was disrupted. By doing a variety of experiments, we came up with this model. So what we found is that in the Shank3 deficient condition, when you lose Shank3, you lost the transport of NMDA receptor to the synaptic membrane through actin filaments. So actin is under control of many regulators. One of the important regulators are RAC proteins. And RAC, by activating PAC and LIMK and the coffilin pathway, they can regulate the stability of those filaments. And when Shank3 was lost, many of these components in the actin regulator families are disrupted. So the actin filaments at the synapse is altered, and then that affects the transport of NMDA to the synaptic membrane. So the Next question is how to rescue those social deficits. A few years ago, there was a landmark paper about the genes that are disrupted in autism. What they found is that there are basically three categories of genes. One is synaptic proteins, such as Shank, transcriptional factors, and the third one is the chromatin genes. Particularly, those histone-modifying enzymes and chromatin remodelers are prominently disrupted. So we think that maybe targeting those histone modifiers will be an effective way to treat autism. So from these established genes that are disrupted in autism, you can see many of them have something to do with actin regulation, actin regulators. And some of those are synaptic proteins, ion channels. And the third group are histone modifying enzymes and chromatin remodelers. So I'll tell you a little bit about epigenetic regulation by histone modification. So basically, histone is the chief component in chromatin. It acts to package DNA. So DNA is wrapped around these histones. At the tail of the histone proteins, there are many modifications that can change the interaction between histone with DNA and then affect the gene regulation. These tails can be either acetylated or methylated or phosphorylated or ubiquinated. These post-translational modification of histones can directly affect gene transcription because it will change the chromatin structure, make it more accessible or less accessible for transcription factors. And these changes of the histone tails are categorized by a lot of enzymes, like histone acetylation that is regulated by histone deacetylase or histone acetylase. Histone methylation is regulated by histone methyltransferase or histone demethylase. DNA methylation is regulated by DNA methyltransferase or DNA methylase. So these enzymes will modify the histone tails or DNA promoters and then make it more accessible by those transcription factors or transcriptional machinery or less accessible. One interesting phenomenon in the genetic screening is that many of the risk factors for autism were found to be actually cancer genes. So there are 43 of them so far that are shared by these two diseases. And about half of them are histone modifying enzymes or chromatin remodelers. So it suggests that these molecules may regulate the transcription and then affect the signaling process in both diseases. Today I will focus on one story that targets histone acetylation. So we know that histone acetylation or deacetylation will directly change the chromatin structure. If it's too much acetylation, it will be the chromatin will be loosened up. The acetylation usually will make it more transcriptionally active. If the deacetylase removes this acetylation, and then it will become inactive because it's more tightly packed, and then it will suppress the gene expression. So we found that in the prefrontal cortical region of the Shank3 deficient mouse, the acetylated level of histone 3 is strongly decreased. And this reduction can be normalized or can be reversed by treatment with a highly potent class 1 HDAC inhibitor called Romidepsin. So Romidepsin is a FDA-approved anti-cancer drug. It has a very high affinity to HDAC, like nanomolar potency to inhibit it. So basically, we give a very low dose of Romidepsin treatment, IP injection, 0.25 milligram per kilogram, which is about 5% dose of the human dose used in cancer treatment. And we inject it once for 3 days. And then this effect can last for quite some time. This was done actually a few days after the injection. And then we looked at the social behavior in the 3-chamber social preference test. And what we found is that the Shank3 mice treated with Romidepsin had a strongly increased social interaction time and a strongly recovered social preference. The Y-type animal treated with Romidepsin had no impairment or no further improvement. The saline treated Shank3 is the same as the non-treated. So basically, it restored the social preference to almost the control level. This is the heat map from the 3-chamber test. You can see that in the Y-type condition, they spend a lot of time interacting with the social stimulus. And in this Shank3 animal treated with saline, not much preference. And after Romidepsin treatment, the social preference is strongly restored. And the Y-type Romidepsin treated has no difference, still preferring the social object. And then we further looked at the duration of the effects. So Romidepsin can rescue the social deficits 1 day or 4 days or 9 days or even 21 days after the last injection. So the effect was very long-lasting. It's basically covering the whole developmental stage for the social skill. So we treated the animal at the age about 5-week-old because at that time it has the robust deficits. Before the treatment, we usually measure the behavior. And then after 3 days of treatment, we start to measure it at different days. So one round, single round of treatment, we can have a long-lasting effect that lasts for about 3 weeks. And not only the social preference index that was increased, but we also found that the social interaction time was strongly increased at all these time points. And these are the heat maps. You can see the strong preference 4 days, 9 days, 14 days, or 21 days after the treatment. Saline injection didn't improve it, no matter how many times you treat it. How many times you test it. It doesn't learn to be social. And we further looked at different doses to see whether it's a dose-dependent effect. We used a 10-fold lower dose, 0.025, and this low dose didn't have a sustained effect. It only gave a transient effect, lasting 1 day. And then we also tested a higher dose, 4 times higher than what we use usually, 1 milligram per kilogram. And it also have a long-lasting effect that lasts at least 3 weeks. In another social test, the social engagement test, we found that Romidepsin can also significantly increase the total sniffing time. So this is a heat map showing the Y-type with a lot of interacting time with the social stimulus, and the Shank3 deficient mice significantly reduced interacting time. And then this is Romidepsin treated, and this is the Y-type with the treatment. So all these 3 are very similar in terms of their social interaction time. To find out whether other behaviors are affected by this treatment, we did a variety of behavior tests. First of all, we looked at locomotion. There's no obvious changes with the Romidepsin treatment. And then we looked at the rotor test to see how well the movement coordination is. They are also fine. There's no obvious deficit and no obvious improvement. And the anxiety-like behavior, the open field test, we see a slight reduction in the Shank3 mice, but not much change after treatment. Another phenotype we have is a self-grooming, repetitive grooming behavior. Shank3 has a strongly increased self-grooming compared to wild type, but this behavior abnormality was not reversed by Romidepsin treatment. The animal still groomed a lot. So far, we have tested one Shank3 deficient mouse model, which is the C-terminal Shank3 deletion. In order to know whether this rescuing effect can occur in other mouse models, we used another Shank3 deficient mouse model that have the N-terminal exon 4 to 9 deletion. And we found that these exon 4-9 Shank3 deficient homozygous mice, they also exhibit very strong social preference deficits. So this is Y type, this is this Shank3 N-terminal deletion. And then after treatment with Romidepsin, we can see the strong rescue of the social preference. And this kind of rescuing effect can be observed in almost every one of these animals. And amazingly, this effect is also long-lasting, about 3 weeks, lasting about 3 weeks. So it's similar to the C-terminal Shank3 deletion animal. Because HDAC inhibitor like Romidepsin has been used in cancer treatment, people think that it may have side effects. So we looked at the cell survival. We found that the cortical pyramidal neurons, which are labeled with CAM kinase 2, alpha CAM kinase 2, had no change in terms of the neuronal density, suggesting that there's no cell loss. Plus, the previous study showing that behavior are also fine, so they didn't have obvious side effects. Then we tested a bunch of currently used antidepressants or antipsychotics to see whether they also can rescue the social deficits in this Shank3 mouse model. So first thing we tried is fluoxetine. We gave the animal 14 days of injection and tested at different days after last injection. We didn't see any effect. So this is a long treatment. And then we tested clozapine. We gave a 3-day injection and tested 1 day or 4 days after the last injection, no effect. And then the third one we tried is the valproic acid, VPA. It's used as a mood stabilizer. It's a very weak HDAC inhibitor. So we used very high dose, like 100 milligram per kilogram, 3 days IP injection. We saw a transient increase of the social preference index. And then this effect disappeared a few days later. We further tested 2 FDA-approved autism drugs, aripiprazole and risperidone. Both drugs were approved for autism treatment, but it's actually just for managing the irritability associated with autism. So both compounds failed to have any effect on the social deficits. So we gave these 3 days injection and then tested at different days after injection. You can see there's no effect whatsoever. And then we further looked at other HDAC inhibitors The first one we looked at is the triclostatin A. That is a highly potent pan-HDAC inhibitor. It induces a transient effect, but only lasts for a day and then disappear. The second HDAC inhibitor that worked similarly to romidepsin is MS-275, also called entinostat. It can rescue the effect relatively fast and then sustain for about 2 weeks or 10 days. So the next thing we need to know is which HDAC is the potential target of romidepsin. We looked at all these class 1 HDACs and we found that only HDAC2 is upregulated in the prefrontal cortical region. This upregulation is happening on the mRNA level and also on the protein level. And then we tested whether knockdown HDAC2 will be able to rescue autism-like behavior like romidepsin. So we designed 2 different shRNA to knock down HDAC2. Both of them strongly decreased HDAC2 expression. They are against different regions of HDAC2. And then we injected these shRNA lentiviruses into the prefrontal region of Shank3 deficient mice or the wild-type mice. And you can see that in the scrambled shRNA injected animal, the wild-type still has a preference, strong social preference, not the heterozygous Shank3 deficient mice. But when we injected these shRNA to knock down HDAC2, these heterozygous Shank3 deficient mice has a strong social preference, similar to wild-type. And the wild-type animals injected with these HDAC2 shRNA had no effect on their social preference. So if you look at the social preference index, you can see the strong recovery by either of these shRNA lentiviruses. This is the heat map showing that when you inject the shRNA lentivirus to the frontal cortex, you can see the strong rescue of the social deficits. So the one question that is puzzling us is that how come when you lose one synaptic protein will lead to the change of a nuclear protein? So we think that there must be something that can shuttle between the synapse and the nucleus. One potential candidate is the nucleocytoplasmic shuttle protein, beta-catenin. Beta-catenin is involved in both cell adhesion and also transcription regulation. We suspect that maybe beta-catenin binds to Shank3, and when you lose Shank3, you lose the synaptic localization beta-catenin, and then you can get to the nucleus. So to prove that, we first looked at the interaction between beta-catenin and Shank3. So this is the IP beta-catenin and western blot of Shank3. You can see this clear interaction between these two. This is the negative control. So beta-catenin actually is one of the binding partners of Shank3. And then we want to see whether after losing Shank3, beta-catenin will be translocated to the nucleus. To prove that, we did the biochemical assay to examine the synaptic beta-catenin, the cytosolic beta-catenin, and the nuclear beta-catenin. And you can see that the synaptic pool of beta-catenin is strongly decreased. And then the active beta-catenin, the cytosol, is increased. And then the nucleus beta-catenin is also significantly increased, supporting our hypothesis that losing Shank3 leads to the translocation of beta-catenin from synapses to the nucleus. To further prove that beta-catenin plays a causal role in regulating HDAC and the social behavior, we looked at the way to, first of all, we looked at the regulation of HDAC2 by beta-catenin. Because when beta-catenin gets to the nucleus, it's potentially able to regulate the genes with the TCF-LEF motif, because they will form a complex with this transcription factor and then regulate the transcription of the target genes. So we found that in the promoter region of HDAC2, there are quite a few TCF-LEF binding motif, and we designed oligo against this region and used a chip assay to look at the binding between beta-catenin and the HDAC2 promoter. We found that the level of beta-catenin at the HDAC2 promoter region, especially the TCF-LEF binding motif, is strongly increased in Shank3 deficient mice, suggesting that the translocated beta-catenin can bind to the promoter region of HDAC2 and then facilitate its transcription. And once HDAC2 is turned on, it will turn off many of the downstream genes. So next thing we did is to prove beta-catenin is a causal factor upstream of HDAC2 regulation and social deficits. So to do that, we first overexpressed beta-catenin in wild-type animal. And then what we found is that HDAC2 is strongly upregulated, similar to the Shank3 deficient condition. And then we looked at the three-chamber social preference, and we found that the wild-type with the overexpression beta-catenin nucleus has actually the social deficits similar to Shank3 deficient animals. The next thing we did is to knock down beta-catenin in Shank3 deficient mice and see whether it will block the upregulation of HDAC2. So you can see that when we use the beta-catenin acid triangle antivirus, we can definitely induce the knockdown of beta-catenin. And meanwhile, we can see the reversal of the HDAC2 expression. Meanwhile, in the three-chamber sociability test, we found that knocking down beta-catenin in the heterozygous mice can actually rescue the social deficits. So these evidence have proven that beta-catenin is sufficient and necessary for the HDAC2 regulation and social deficits. So the next question is, what are the downstream molecular targets of romidepsin or HDAC2 inhibition that may be important for the synaptic deficits or behavior deficits? So the first thing we looked at a few glutamate receptor subunits. We found that only NR2A subunit mRNA strongly increased after romidepsin treatment. The Shank3 is not rescued by romidepsin treatment. It's not only happening at the mRNA level, but also at the protein level. You can see this is the NR2A subunit that is not changed in the Shank3 deficient case, but is elevated by romidepsin treatment. The Shank3 was lost in Shank3 deficient mice, but cannot be rescued by romidepsin treatment. So to find out whether the upregulation of NR2A by romidepsin treatment has something to do with histone acetylation, we designed several primers against the NR2A or NR2B promoter region, and then find out whether the histone acetylation level at this promoter region have any change. We found that NR2A subunit has a strong increase regarding the acetylated level of histone 3 at its promoter, not NR2B. So this is consistent with the upregulation of the transcription of NR2A by Romidepsin treatment. Then we further did electrophysiological studies to look at NMDA-mediated synaptic function. We found that in Romidepsin-treated Shank3 mice, the NMDA receptor-mediated EPSC restored to almost the Y-type level. Compared to the saline-treated one, it's significantly different. AMPA EPSC has no change between Shank3 deficiency or Romidepsin treatment. And this kind of recovery also has a time, it also time dependent. This was done a few days after injection, and this was done 16 to 18 days after injection. You can still see the rescue, but not 30 days after. So at this time, the behavior rescue also disappeared. This is the time this behavior rescue is still present. So this is very well correlated. We further knocked down HDAC2 and then tested whether it can restore the NMDA function. And indeed, it can. So this is a SHRA lentivirus injected. And fluoxetine, which is the antidepressant that failed to rescue behavior, we didn't see any rescue with the NMDA EPSC. And then our previous studies know that several actin regulators are disrupted in Shank3 deficient mice. So we looked at their transcription after Romidepsin treatment. We found that beta-PIX, which is the RAC1 activator, has a strong loss in the Shank3 deficient animal, and this can be restored by Romidepsin treatment. The LIMK, which is downstream of RAC, is also decreased in Shank3 deficient mice, which can be restored by Romidepsin treatment. Other actin regulators, we didn't see the strong differences in their mRNA level. So we further looked at a few other risk factors that are in the interactome of Shank3, such as cortectin, cortectin binding protein, ANC2, and SYNGAP. Only cortectin binding protein and ANC2 has a strong upregulation by Romidepsin treatment. So these potentially will help to regulate actin filaments. At the protein level, we can also see the rescue of beta-PIX and LIMK and the upregulation of cortectin binding protein and ANC2. We further looked at the acetylation at these actin regulators by designing primers against their promoter region. So we found that beta-PIX can be upregulated by Romidepsin treatment and also LIMK. So this is the acylated H3 level at the promoter region of different genes. Because these actin regulators are either restored or elevated, we try to find out whether actin filaments can be rescued. So we found that in the Shank3 deficient animal, the actin filaments in the synapses of prefrontal cortex are strongly decreased, not PSD95. And then after Romidepsin treatment, it's restored to the Y-type level. Romidepsin treatment of Y-type had no change. So this is the F-actin level, and this is the PSD95 level. No change by Shank3 deficiency or Romidepsin treatment. To find out whether there are additional genes that are dysregulated by Shank3 deficiency and restored by Romidepsin treatment, we performed the RNA-seq analysis to look at the genome-wide restoration. So you can see that there are actually about 200 genes that are downregulated in the Shank3 deficient animals. Those are saline-treated Shank3 deficient animals compared to wild-type animals. And after Romidepsin treatment, you can see that most of them are restored to close to the control level, to the wild-type level. And when we look at those genes, many of them are related to actin filament-based process, cytoskeleton-dependent transport. And some of them are related to receptors, signaling molecules, signal transduction. And the third group are related to development. So this is consistent with what we found, because when you treat adult animals with Romidepsin, it has very limited effect. So it must have some developmental process or molecules that are crucial for the rescue effect. There are certain genes that are not changed in Shank3 deficiency, but can be elevated by Romidepsin treatment. These genes are enriched in cell-cell signaling, synapse transmission, G-protein coupled receptor signaling, etc. So in the end, based on these studies, we have kind of a model that may explain why Romidepsin potentially can treat the social deficits in autism. When you have Shank3 in the synapses, they have a very well-organized complex, and then the actin filaments are regulated by these genes, and NMDA receptor can be transported to the synaptic membrane, and then the animal shows normal social behavior. When Shank3 is lost, beta-catenin is nuclear translocated and binding to the promoter region of HDAC2, turned on HDAC2, and then this will lead to the silence of many of the downstream genes, including the actin regulators, and then actin filaments are disrupted. When you inhibit HDAC2, you will be able to restore these downstream genes. And then the actin filaments will be restored, and the NMDA receptors can be delivered to the synapse. So you have the normal synaptic function and normal social behavior. So this work is the product of a teamwork. Dr. Cheng has done a lot of biochemical and electrophysiological studies. Kai Jie did many of those behavior studies, and Zhen also helped. Oh, she's not on this picture. On the chip assays, and Dr. Hu did the RNA-seq analysis. Laura did the original studies on the Shank3 mice characterization. So this work is supported by Nancy Laurie Marks Family Foundation and NIMH. So after we published this paper on Nature Neuroscience, I have received many emails from patients or patients' parents. They basically want to know whether there's any clinical trials that can be their kids can participate. At this stage, it's a little bit early, and we will work hard to translate our preclinical studies to clinical settings. But I received an interesting email from Mark McCarthy from San Diego. Basically, he told me that there's an endogenous HDAC inhibitor, B-Hydroxybutyric, and this endogenous HDAC inhibitor can be generated from a ketogenic diet. And there are two small case studies showing that ketogenic diet has a very significant effects to improve the social behavior in autistic children. So this is the structure of a beta-hydroxybutyric acid. It can cross BBB into the central nervous system. The ketogenic diet basically is a high fatty acid diet. It has been used for treating epilepsy in children. And it was found that the beta-hydroxybutyric can suppress oxidative stress, increase BDNF level by inhibiting class 1 HDAC. And the significant improvement of sociability in autistic children in these case studies by ketogenic diet, even though the mechanism is unknown, probably it could be through the inhibition of HDAC1. Class 1 HDAC. Thank you for your attention.
Thank you so much, Professor Yan. We do have a couple of questions that we can go through now. One question that we had is the conclusion that NR2A NMDA trafficking is the causal issue in this model, and that Shank3 insufficiency itself is not the mechanism for social deficits?
Okay, yeah, it's a great question. Actually, I think it's very difficult to say what is exactly the molecular changes that cause social deficits. We think that NMDA receptors play a key role in this case because when we block NMDA receptors in prefrontal cortex, we actually can see the social impairment. Shank3 is a master control gene in the synaptic postsynaptic side at the synapses. It interacts with NMDA receptors. So when you lose Shank3, you affect NMDA receptor function that may contribute to social deficits. So Shank3 could be a kind of trigger the loss of Shank3. Yeah, but Shank3 has to work through other proteins that interact with Shank3 in order to pass the signal between neurons. So NMDA receptors, I think, plays a very important role in regulating social behaviors.
Another related question is, are you concerned that reestablishing NMDA receptors to the postsynaptic membrane in a synapse with reduced Shank3 might interfere with learning, a process that might be very dependent on having the normal Shank3 in the postsynaptic density regulating NMDA?
Okay, yeah, it's a great question. We actually didn't see the interference of NMDA receptor by Romidepsin treatment in wild-type animals. So if you look at the physiological studies, you can see that it restored. So this is a Romidepsin treatment in Shank3 mice, and it restored the NMDA response to the control level. For the wild type treated with Romidepsin, it didn't have any alteration. So we don't think that it will impair anything in the wild type. We also did those behavior studies, and we didn't find any obvious abnormality in wild type animals.
Another question that we had was, do you think that these findings are going to be Shank3 specific, or do they suggest that HDAC inhibitors might be involved in autism more generally?
Yeah, this is a good question. Again, even though we used Shank3 mouse model in this case, we think that targeting histone modifiers may be applicable as a treatment for a variety of autism conditions. Because from those genetic screening, you can see that many of those histone modifiers are changed in autistic brain from those human patients, and they may have Shank3 deficiency or may not. Right? The small case studies with the ketogenic diet on these autistic children, they actually probably don't have any Shank3 deficiency. So this study is like containing 45 kids. This one have 15 kids. So these kids are just autistic kids. I don't think they have Shank3 deficiency.
Another question that we had was about the phenotypes for the Shank3 mutants that you use in the paper. The question was, a previous work using these mutants seems to find discrepancies in the social phenotypes that you find. How do you account for these discrepancies?
Okay, yeah. So Shank3 has many different regions, like PDZ domain, SH3 domain, C-terminal, N-terminal. So different mouse models have been generated against those different regions. So just for C-terminal deletion, we found very strong social deficits, even in the heterozygous mice. There was a previous study using similar mice, and they didn't find such a strong social deficits. There are a couple reasons for this discrepancy, potential discrepancy. First, there are many differences in terms of the way the behavior was carried out. So, for example, we used this kind of way to measure. We habituate the animal with the empty capsules present in the apparatus. So when the animal is tested with in these phases, they are not distracted by these capsules. Secondly, our apparatus is bigger, so the animal will have to actively engage into the in the interaction in order to be counted. And the third is that we are only using males. We found that females do not have the same deficits. And there's a European group, they actually also use the C-terminal deletion animal, and they found very strong social deficits. It was published in 2016 on Science. So I think there are some consistency there.
Another question we have is, the comparisons seem to be against wild type, but is it possible that there is non-specific effect of Romidepsin that would work across other genetic mouse models of autism that do not work on the same HDAC system, and that an alternative mechanism is at work? Has the same approach been taken comparing against non-wild type controls?
Can you repeat the question again?
The comparisons seem to be against wild type, but is it possible that there is a non-specific effect of Romidepsin that would work across other genetic mouse models of autism that does not work on the same HDAC system, and instead there's an alternative mechanism at work? Has the same approach that you take here been taken comparing against non-wild type controls?
So in this study, most of the comparison was the Y-type littermates, and it's possible that different mutations related to autism can be treated by targeting different histone modifiers. I'm not saying that HDAC inhibitor will be the magic bullet that will work in every case. We don't have the evidence now, but we are looking into it. We are looking at other autism models and see whether Romidepsin is still able to work. And I think it's probably one of the studies that may lead to further interest in looking into the possible treatment strategy by targeting histone modifiers. Whether there are some non-specific effect, we have done those HDAC lentiviral experiments to rule out the possibility. So basically, we just knock down HDAC2 in prefrontal cortex, and we can see the similar effects as Romidepsin injection into the whole body. And we also looked at those target genes. We didn't see the same change in peripheral organs or other brain areas, such as striatum or VTA.
And finally, just the last question, then we'll have to wrap up there. Is there any evidence that beta-catenin nuclear localization is synaptic activity dependent?
Beta-catenin activity can be regulated by Wnt signaling, so or location can be regulated. So maybe synaptic activity can regulate beta-catenin activity. It is possible. Yeah.
Okay, great. I'm afraid that we need to wrap up there. Thank you again, Professor Yan, for your presentation, and thank you everybody for tuning in. A complete replay of the Journal Club will be available on the Spectrum News site in a couple of days, so check back there to get the full presentation. This Journal Club is part of an ongoing series, so please check our site for upcoming talks to be announced soon and for video archives of previous webinars. Our next webinar is with Feng Zhang, and that's on June 6th. And thank you again, Professor Yan, and thank you all for tuning in.
Yeah, thank you.